Hello and welcome to Neurosurgery Written Board Crash Course. My name is Chen. We're going to talk about visual pathway. The visual pathway is comprised of different units listed here, and they're listed in the order in which the signal is transduced. You can see here that the signal light first enters the eye in the retina, that then transmits signal to the optic nerve, chiasm, the optic tracts, that then goes to the lateral geniculate nucleus, that then becomes the optic radiation and eventually transmits the signal to the primary visual cortex, which is where the conscious vision is located. There are other visual pathways that will not be covered in this video, and namely those are the pretectal area, which is responsible for the light reflexes, the superior colliculus, which is responsible for the eye movement reflexes, and the suprachiasmatic nucleus in the hypothalamus, which is responsible for the neuroendocrine function. So let's start from the beginning. The retina is comprised of several cell types, and the most important ones are listed here. As the light enters the back of the eyeball, it activates the rods and cones, which are photoreceptors, and these are not actual neurons. The rods and cones then conducts signal to the bipolar cells, and these are actually primary sensory neurons. And these primary sensory neurons then transmit signal to ganglion cells, which are considered secondary neurons. The ganglion cells assembled together to form what we know as the optic nerve. The figure on the right shows a schematic of the retina, and the OPL stands for the outer plexiform layer, the IPL stands for the inner plexiform layer. The on and off denotes the part of the IPL where the circuits responding to the onset and the offset of the light, respectively. And we'll cover the on-center and off-center, or the onset or offset, in a little bit. The retinal ganglion cells can be further subdivided into different classes. And the classification is different based on the different criteria you use based on morphology or the response to light. The ones that we will be concerned with is the X, Y, and W system. The X ganglion cells is comprised of the great majority of the ganglion cells, and they're concerned with the analysis of detail and color. The Y ganglion cells detects motion, and the W ganglion cells project to the brainstem. And not a whole lot is known about these ganglion cells, and so you probably don't need to know a whole lot for the board exam. The table on the bottom shows the different retinal ganglion cell types and their differences. I don't really have a good way to remember the whole table, but in terms of the response type, I'd like to think of that there are only 3 places that the ganglion cells can respond to. And namely, those are LGN for conscious vision, pretectum for the light reflex, and the superior colliculus for eye movement reflex. I like to think of the superior colliculus as the kind of the responsible for the upgaze palsy and the Perinault syndrome. Now the pretectum and the superior colliculus will be covered in other videos. So each of these ganglion cells will project only to 2 places, but there are 3 places to project to in total, and so they have to play a little bit of a musical chair. And so in the case of W ganglion cells, we know that it projects to the brain stem, and so therefore it has to project to superior colliculus and the pretectum because of their location. In the case of Y cells, because we know that it's responsible for motion detection, we know that In order to detect motion, we have to see the stimuli and then move the eyes to detect motion. And so therefore, it needs both the LGN and the superior colliculus for both conscious vision and the eye movement reflex. And that leaves us with the X cells, the pretectum and the LGN as the only two that haven't been used twice. So now let's talk about the ganglion cell receptive fields. The receptive field for a particular ganglion cell can be thought of as a group of photoreceptors that connects to that one particular ganglion cell. And the different ganglion cells will have different photoreceptors connected to the other ones, and therefore will have different receptive fields. You can think of this as another way as the region of visual field in which light stimuli can evoke a response in that particular ganglion cell. And within each receptive field, it can be subdivided into two regions, a center and a surround. And we will cover that shortly. So each receptive field has a center and a surround. The center of each such field receives a direct connection from a small number of photoreceptors of rods and cones, and in this figure is highlighted by the red circle. While the space in the surrounding area, conveniently called the surround, will receive input from a larger set of photoreceptors whose activity is relayed by the horizontal cells in the retina. The light shining on the center of a bipolar cell's receptive field and light shining on the surround produce opposite changes in the cell's membrane potential. The diagram here uses an on-center with an off-surround bipolar cell as an example. When monitored at rest, there is a tonic baseline firing at rest when there is no light. If light is shined on the center of this cell's receptive field, this excites the ganglion cell, causing it to emit action potentials at a higher frequency, as you can see in this example in the red box. Conversely, if light were shine on the surround of the receptive field, it will inhibit the cell's response by decreasing the firing rate. And if you have a light that shines directly on both the on center and the off surrounds, it will remain its baseline firing rate because the excitation of the on will offset the off in the surround. Now in contrast, there is another kind of bipolar cells, the off-center on-surround, has the exact opposite effects of what we just talked about. This type of cell becomes depolarized when an area of darkness strikes the center of its receptive field, and hyperpolarized when it strikes the surround. So let's move on to the optic nerves, chiasm tracts. Here's a diagram that shows the different defects that can be obtained from lesions at different locations. So I won't belabor the point here with each individual one, but I just wanted to cover one specific lesion that is very intriguing. It's called the junctional scotoma. So as the optic nerves merge into the optic chiasm, note that immediately after crossing, the inferior nasal loop forward for a short distance into the optic nerve of the opposite eye in this place, this location is called the anterior von Willibrand's knee. A lesion at the anterior chiasm will cause what is known as the anterior chiasmal syndrome. A lesion at the anterior von Willibrand's knee affects the ipsilateral optic nerve fibers and contralateral inferal nasal fibers. The resulting deficit is typically called the junctional scotoma, which is a combination of central scotoma or blindness of the ipsilateral eye plus a superotemporal quadrantenopsia of the other eye. Typically, I like to think of this as blind in the ipsilateral eye plus a pie in the sky deficit in the contralateral eye. The optic tracts end in the lateral geniculate nucleus, the LGN. There are 6 layers in the LGN. The first 2, layers 1 and 2, are called magnocellular layers because they contain large cells. The rest of the layers, 3 and 6, are parvocellular layers because they have smaller cells. And the parvocellular system is concerned with color and detailed form, while the magnocellular system is concerned with location and movement. The fibers from each eye go into the LGN in separate layers. The axons from the contralateral eye of the nasal hemiretina will project to layers 1, 4, and 6, whereas axons from the ipsilateral eye, the ipsilateral temporal hemiretina, will project to layers 2, 3, and 5. So there is no crossover between the eyes in the LGN at this point. Now, once they have synapse in the different layers in the LGN, the postsynaptic axons will emerge from the LGN to go to the primary visual cortex as the optic radiations. And at this point of the visual cortex or visual processing pathway, the LGN still contains the on-center off-surrounds or off-center on-surrounds for the visual processing. So the optic radiations is also known as the geniculocalcarine tracts. We all know that the optic radiations will travel from the LGN to the primary visual cortex, aka the calcarine fissure, in different ways. And one of them is depicted here, called the Meyer's loop. It detours entirely around the tip of the temporal horn and passes posteriorly around the lateral ventricle and converges on the lower lip of the calcarine fissure. And note on the bottom left where it depicts the Meyer's loop genu and its relationship in the temporal horn, and hence its importance in knowing the anatomy of this structure when it comes to the temporal lobectomies. The other structure is called the Baum's loop. It travels straight from the LGN up and around to the occipital cortex via the parietal lobe, and it ends in the superior lip of the calcarine fissure. Now this picture depicts the visual pathway in a slightly different fashion. The superior is located on the top and the inferior is located on the bottom, and the visual processing pathway goes from the left to right. Now as light from the superior quadrants enter the inferior retina, signal travels to the LGN, then to the Meyer's loop, and subsequently project to the inferior bank of the calcarine fissure. And notice how the light from the superior quadrants stays on the bottom of the visual pathways, and this is and therefore the lower part of the brain of the calcarine fissure sees the superior visual field, and this is basically the basis of the pie in the sky deficit. Now conversely, light from the inferior quadrants will enter the superior retina, goes to the LGN, and then travels to the superior trajectories of the geniculo-calcarine tracts, and then projects to the superior bank of the calcarine fissure, and therefore the top of the brain sees the inferior visual field. Now, the axons conveying the information from the macula, or the fovea, or the center of the visual field, arise from the central portion of the LGN, and it synapses on the caudal pole of the occipital cortex. The macula is disproportionately represented in the visual cortex, and it takes up roughly 1/3 of the calcarine cortex for that very small area of the retina that it covers. The optic radiation eventually ends in the visual cortex. The primary visual cortex is located in the calcarine sulcus, and more precisely it is located in the medial surface of the occipital cortex, in the depth of the calcarine fissure. It extends as far posterior as the occipital pole and as far anterior as the parietal occipital sulcus. The cuneus gyrus lies above it, and the lingual gyrus lies below it. And then we have the secondary and tertiary visual cortex that are directly above it and below it. These are the association cortices that are responsible for integrating visual signals. Now recall the band of genari, or the striae of genari, that was covered in the neocortical layer video, and you can find more information about this in that video. But in short, it is basically a myelinated fibers of the optic radiation that enters this area in layer 4 of the gray matter cortex. And this is what gave the visual cortex its striated appearance on brain sectioning. Now let's talk about visual processing in the cortex. Now recall earlier that we talked about the retinal ganglion cells, the center surround receptive fields, takes on a circular shape. That's depicted by the pictures on the top left. The visual cortex forms its own receptive fields by combining the individual on-off centers to form a line detector of different orientations depending on which receptive field you combine. So the cortical regions, as a rule, are organized into functional columns. These are the basic unit in which information processing happens. Now, in the case of the primary visual cortex, the functional columns are also called the ocular dominance columns. There are two types of the functional columns in the visual fields in the visual cortex based on what information it receives. Now, the cells in one functional column receives afferent only from the LGN, lateral geniculate nucleus layer 2, 3, and 5. Now, if you recall from earlier, these layers brings information from the ipsilateral eye, and hence these functional columns are considered ipsilateral functional columns. Similarly, an immediately adjacent column receives afferent from the lateral geniculate nucleus layers 1, 4, and 6, and those receives information from the contralateral eye, and therefore are considered the contralateral functional columns. And so, as a result, the origin of the visual information alternates from left to right eye across the functional columns. And so, a pair of the left and right ocular dominance columns corresponding to the same spot in the visual field is called a hypercolumn. So each of the hypercolumns are organized across the primary visual cortex in the retinotopic map that represents the visual fields, and as it depicted by the pictures on the left. Now this is basically just another way of saying that each point in the brain has a corresponding spot in the retina, and therefore the representation of the outside world is mapped onto the primary visual cortex. Now across a hypercolumn, there are different possible angles or orientations of the bar stimulus in the visual field, and these are called the orientation columns. And later on, once the information has been processed in the primary visual cortex, information will be passed on to the association cortices and other cortices for higher level of processing. These are my references. I hope you find this helpful, and we'll see you next time.